We are ready to look at our next type of saccharide, and it's our polysaccharides. So we looked at monosaccharides and disaccharides. We looked at those sugars, and now we're going to get into where there are many of our glucose molecules. So we're going to stay focused on glucose, okay? We're going to look at that, but we're going to look at what happens when you link many of them together. So let's see, what does poly mean? It means many. So as I said, this is many saccharides that are linked together. How do we get polysaccharides? Well, plants are bringing in carbon dioxide, they're taking in water, and they are converting that carbon dioxide and water to glucose. Plants are doing that, and when they take those glucose molecules, they link them together many times over. Okay, these are what produce for us starches that we consume for calories and cellulose that we consume for fiber. Okay, so let me move over to the screen because most of what I'm talking about right now is over there, and I can point to various items here. When we are dealing with these polysaccharides, we use the word complex carbohydrates. You've heard that when people talk about a diet needing complex carbohydrates. That would be as opposed to simple carbohydrates, which are our sugars. Now, when we consume starch, these are the digestible ones, we can actually get the glucose molecules out of the starch when we consume them. This is not true for the cellulose. It is what when we consume it, it's still very important for our diets. It is what we would call dietary fiber. So we can consume this starch, the cellulose all day long and not get any calories from it, even though it's made up also of glucose molecules, simply because we are unable to digest them. So let's focus in on the starch, okay? It is a major food reserve that the plants have, okay? So this is what the reserve of food is. You've got certain plants that are very high in starch. When you eat a diet that has a lot of corn, rice, potatoes, your wheat, your breads in other words, oats, rye, and barley, we consume these to get our starches. We'll get calories from that, from the complex carbohydrates of starches. Unripened fruit is also high in starch, okay? If you've eaten an unripe banana, it doesn't taste very sweet. But as it ripens, a fruit will typically hydrolyze. Remember what that means? It will break apart into the sugar. So it'll break those bonds between the glucose molecules and convert them to very typically, our fructose, maybe our glucose, maybe sucrose, depending on what the sweetness is. Now, when we consume a starch, okay, it is broken apart. That is what depolymerize means. Starch is a polymer, repeating pattern of these glucose molecules. So if it's depolymerized, it's broken apart, and it's broken back to the monomers of glucose. So starch is made up of a bunch of glucose molecules that our body can use. Now, when we take in starch, if we were to leave it in our mouth, this breakdown process will occur. Nobody wants to leave a cracker in their mouth until this happens. We chew it up, we swallow it. But if you want to do an experiment, you can put a piece of bread in your mouth and the saliva will start breaking it down, and you can start tasting the sweetness of it as that process takes place. There are two main types of starch. There's amylose, and amylose is a chain of glucose, just one glucose molecule after another glucose molecule. We'll see an image of this in just a moment. Oh, there it is. So this is representing our glucose molecule right here, okay? So that's representing our glucose molecule. It's linked by an oxygen bridge, and that's what I'm going to call this right here. This is our oxygen bridge. We're going to examine that oxygen bridge as we continue. Now we've got just one glucose molecule after another glucose molecule to make amylose. And now I've written right over top of that, so let me erase it. Amylopectin doesn't have just a straight chain. It has a chain of glucose molecules, but also branches that come off of it. So here is our amylopectin. Amylopectin, so you have a chain of glucose happening, but coming off, there's lots of oxygens on those glucose molecules, aren't there? So we could have another chain occurring here where they are connected. So we've got chains that have bridges between them. So most of our starch, it says here 80%, most of our starch is amylopectin. Now let's mention cellulose for a little bit. So that's your plant fibers, okay, is what cellulose is. It is, and I find this a remarkable statement, it is produced in greater abundance than any other substance on the planet. We could have a chemical plant pumping out plastics as fast as they can, but it's not produced at near the abundance of what cellulose is, because every plant out there, as it grows and as it lives, is producing cellulose as it grows. So it is also a linkage of glucose molecules. So there's got to be something different if we can digest one and not the other. Now we need to look at why cellulose and why starch are different, and why one we can digest and one we cannot. Beta linkage, and we're going to look at this here in just a moment. Beta linkage is what we find in cellulose, and we are not able to break that bond between the glucose molecules and digest that. We need certain enzymes to do this. Now, what is a beta linkage? A beta linkage starts with a beta D-glucose. So let's just examine this glucose molecule right here, okay? And this molecule is if we were to consider it being a beta, it would have an OH group above the ring, right? Because betas look like butterflies, and butterflies fly in the sky. So we have that happening there. At carbon number 4 on the neighboring ring, the OH is always below the ring. So here is the OH below the ring. We take off an H and we take off an OH and we allow a linkage to happen. So it is going up from the first glucose ring into the bottom of the second glucose ring. And that is what you call a beta 1, because that's you're starting on the left side, to the 4th carbon on the next ring. Every one of these is a beta 1,4 linkage. This gives a very fibrous structure to this molecule. It is in a nice straight line. And if you think about the fibers in a celery stalk, okay, but all of our cellulose are these strands that go into a nice straight line. So that is a beta 1,4 linkage right there that we have in our cellulose. In starch, it's an alpha linkage instead. So we're going to use alpha glucoses instead of beta glucoses to come together. We can digest this. We have the enzyme needed to break the bond in a beta linkage. So, an alpha linkage. So here is our alpha linkage. So the big difference is at the number 1 carbon. We know that at the number 1 carbon, the OH is below the ring, okay? And alpha, it looks like a fish. It swims down in the sea. So we have it down below the ring. But at the 4, it is still also below the ring, okay? So they're both below the ring. That's always true at the number 4 carbon, and it's only true at the number 1 carbon if it's an alpha glucose. So if we take an H off, okay, and an OH off, so we're pulling out the water to link these together, we end up having it come down out of the bottom of this and then going up into the bottom of the other. We have the enzyme. So to me, it is quite remarkable that these molecules are exactly the same except at the oxygen bridge, okay? We're going to call that the oxygen bridge. Now, we're going to learn the difference of this, and we're going to look at the models. And we're actually going to, in class, build a strand of beta 1,4 linkages, which would be our cellulose. And we are going to build then a strand of starches using a bunch of glucoses in the alpha formation so that we can actually see the nice linear relationship that these have and what happens with the starch as opposed to the cellulose in the way the molecule unfolds. Now, why is dietary fiber important? If we can't consume it for calories, why do we need it? So it is going to be just as important as calories, and far, far, far too many people don't get enough in their diet, okay? It is very useful to get your waste out of your digestive system. There's a lot of things in there that we do not want to linger in our large intestine. So it is this dietary fiber that helps go ahead and move all of this through our digestive system. And a diet that is very low in dietary fiber, and if you are just a potatoes person, you like your potatoes and breads, you're not getting enough dietary fiber, and you are susceptible, much more than those who do consume a good diet of fiber, to colon cancer. So how do we get a lot of good fiber? Well, you need things that have bran in it. So if you have whole wheat as opposed to just white bread, you're getting some of that bran. If you are somebody who eats skins, now I'm not recommending you eat the banana peel, but you can if you want. But if you eat the potato skins, you eat the apple skin, they are full of fiber. Any fruit and vegetable are going to be rich in fibers. Now, there is another, that this is not plant produced, another polysaccharide that we need to consider, and that is glycogen. Glycogen is also a highly branched polymer of, it is a branched polymer of glucose. Where did we see that before? We saw that in amylopectin. Amylopectin had branches, but in glycogen, it is highly branched, okay? We have lots and lots of branches, and the branches have branches, but it is, look at the way that this is drawn, down and up. That is our alpha 1,4 linkage. It is, our body is able to break that down. So what is glycogen? It is what we use in our body to store excess glucose. If you consume more calories than you need, the first source of storage of those calories is in the form of glycogen, okay? Glycogen is used for short-term storage because your body can use it rapidly. Once it gets stored, if you need those calories, you can store them, get them out and use them fairly rapidly. Okay, often stored in the liver and also in muscles where it could be utilized for the energy right away. You might have heard of people carb loading before a long distance race or some sort of strenuous exercise. They are hoping to put a lot of glycogen around their muscles so that their body, when it's done using the sugar in their bloodstream, is able to retrieve those calories rapidly. So it is a hydrolysis process. You're putting in water in order to break that bond. So remember, when you break this bond, you have an H and an OH. One of the H's will link up with that oxygen, and then the OH will link up at that carbon, and now we will have broken that bond between them. So if it takes a lot of water to break that down, one thing that I always found interesting is if you are on a diet and you're losing weight rapidly, especially if you're on a low carbohydrate diet, a lot of times it's because your body is just depleting the glycogen that's in there. In order to deplete the glycogen, you have to be putting in water to break those bonds. So H2O has to go in to break those bonds. So when they say you've got a lot of water weight that you are losing at the beginning of a diet, it's as you're depleting that glycogen. But you must deplete your glycogen before it's going to go into the long-term storage and start taking out the fat, which is your long-term storage. Okay, so that is what this is saying. Glycogen requires water to break apart the glycogen and get it into the form of glucose. And we know that the name of that process is add water, break apart. Hydrolysis causes a lot of that water weight to be lost. Now, if you can't store anymore, we know it'll store it first as glycogen, and then when you use up that, or you have stored all the glycogen your body will store, then the rest of it gets stored as fat for long-term storage, for if you were to need it after you used up all of your glycogen. All right, so those are our polysaccharides. We have to make sure we can recognize those polysaccharides. We've got our starches, which is amylose and amylopectin. We have our cellulose. We understand the difference in how they're linked and how we can consume them and use them in our body. And then we have our animal-produced polysaccharide, which is your glycogen. So make sure you can recognize those, and we will play with the models in class to see how they link together.